Hi, my name is Carolina Titus, and I'm a senior in the Mast Research Institute at Sarasota High School. My project is testing the effects of the prolonged exposure of Karenia brevis on the filtration rate of Crassostrea virginica. 2,000 tons of dead marine life was produced by Florida's red tides in 2018 alone, according to the Florida Fish and Wildlife estimates. On top of that, $8 million was lost from Florida businesses in the same year. The price tag on cleaning up beaches from the 2018 red tides reached nearly $100,000 in Sarasota alone. Now, before diving into my research on red tide, let's review some terminology. Karenia brevis is a scientific name for the organism of red tide. These single-celled organisms are dinoflagellates that use photosynthesis and asexual cell division to reproduce. Populations of K. brevis grow and spread rapidly with a combination of three factors: the presence of K. brevis in the water column, a major nutrient source, and a wind or water current to move the dense clusters. The major problem with red tide blooms on our coast is the release of brevetoxins, which are a collection of neurotoxic compounds. These brevetoxins released during a bloom cause many fatalities among different species of fish, birds, and even marine mammals. Moving on, the term Crassostrea virginica is the scientific name used for the eastern oyster. This is the most common species of oyster found in waters along Sarasota, Florida. This species of filter feeder was selected to test the effects of red tide on the filtration ability of our local oysters. Red tide is what is known as an algae bloom, where algae rapidly accumulates in a water column. Red tide poses a major threat to human health, marine ecosystems, coastal aesthetics, and our economy. As previously mentioned, the formation of red tide requires three basic factors: the presence of K. brevis in the water column, a major nutrient source, and a wind or water current to move it. Aquatic organisms can be exposed to brevetoxins in the water in a variety of ways, including direct contact with the toxins in the water, ingesting cells, inhaling toxins, and consuming toxic prey. Toxins then bioaccumulate from producers to consumers through the food chain. This has led to my investigation on the effects of these blooms in bivalves and filter feeders. This experiment tests the effects of prolonged exposure of Karenia brevis on the eastern oyster sea virginica. As I said before, the purpose of this experiment is to test the effects of the prolonged exposure of red tide on eastern oysters and observe the following implications. Bivalve responses to toxic dinoflagellates are species-specific, depending upon algal species, algal toxicity, algal concentration, history of exposure, and differences in digestive function. Testing on C. virginica will provide results on how our local oysters are specifically affected. Also, these blooms of K. brevis pose a serious threat to the shellfish resources and the growing shellfish aquaculture economy in Florida. This recurring issue is in need of a solution. Finally, the gap to be filled in this area of research was to understand the effects of K. brevis on the filtering ability of C. virginica in a laboratory environment. The research done in this experiment addresses this gap and may be an important preliminary study in a laboratory environment that may be continued on a larger scale. My hypothesis is when Crassostrea virginica is exposed to Karenia brevis for an extended period of time, the filtration rates will be lower than the oysters not previously exposed. To start, I collected oysters from the water surrounding Mote Marine Laboratory. The lengths of each oyster were measured, and oysters of average size were selected for experimentation. Oysters were carefully separated from one another and cleaned by hand to remove any algae or additional organisms. 12 oysters were then divided into two separate 4-gallon tanks, 6 oysters in each tank. The oysters were given 24 hours at room temperature with light aeration to assess their health. The oysters were starved for 48 hours prior to experimentation. This ensured that the oysters would begin filtering as soon as Karenia brevis was introduced to the water. The treatment tank was then dosed with K. brevis twice daily, at 9 a.m. and again at 5 p.m. Cell concentrations of the culture of K. brevis were taken before dosing, and dose volume was determined using the dilution equation to reach the first day cell exposure cell concentration, goal of 1 million cells per liter. This count was chosen to represent that of a high to intermediate cell concentration of a red tide bloom, mimicking those that frequently affect the Gulf Coast of Florida. The diagram set up to the right gives an idea of what each tank looked like through experimentation. Once the tanks were initially dosed with Karenia brevis, a sample of the water was collected to confirm the cell concentration. After dosing, a cell count was taken at T0, and samples were then collected after 30 minutes, 1 hour, 2 hours, and 3 hours. After 1 day of exposure, 2 oysters were removed from both the treatment tank and the control tank and placed in separate 1-liter beakers. Each beaker was then dosed with a concentration of 1 million cells per liter to determine the oyster's clearance rates. This allowed me to compare the clearance rates between oysters that had not been exposed to Karenia brevis to those that had been exposed for 1 day. The clearance rate was calculated using the formula from Riskard, shown at the top right. The treatment tank was dosed again the next day at 9 a.m. and at 5 p.m. with an adjusted cell concentration for the remaining 4 oysters in the treatment tank. 2 oysters were once again removed from each holding tank after 2 days of exposure and placed in separate beakers to determine clearance rate. The cell concentration was once again adjusted for the remaining number of oysters. The treatment tank was dosed again the next day at 9 a.m. and at 5 p.m. After 3 days of exposure, the remaining 2 oysters in each holding tank were placed in the beakers to determine their clearance rate. Figure 1 represents the average filtration rate, or the number of cells filtered. The graph shows an overall decrease in each trial, with a slight spike at the end of day 1 of exposure, with a slight spike at the end of 1 day of exposure and 3 days of exposure. This increase in algal concentration at the 4-hour mark for the treatment group may indicate if the C. virginica had stopped filtering or feeding. The decrease in cells per liter over each hour shows the amount of K. brevis cells decreasing as the oysters are filtering the water. The control seems to filter the most cells from the water, likely because it has not been previously exposed to K. brevis. The control shows the highest decrease in algal concentration, starting at 920,000 cells per liter and decreasing to 260,000 cells per liter by hour 4. The difference in K. brevis concentration between 1 day of exposure, 2 days of exposure, and 3 days of exposure does not vary significantly, but shows a higher K. brevis concentration compared to the control, which started at about 900,000 cells per liter and only decreased to an average of 540,000 cells per liter. This shows the C. virginica previously exposed to Karenia brevis had lower filtration than when re-exposed in experimentation. Figure 2 represents the average clearance rate, or the volume of water filtered. The graph shows an overall spike in the beginning for each trial. This may have been when the oysters first started filtering. They were working efficiently. Following this initial spike, 1 day of exposure, 2 days of exposure, and 3 days of exposure decreased. This is likely because of their prolonged exposure to K. brevis and the toxins affecting their ability to filter. While this slight decrease may not be significant, the data indicates that previously exposed oysters have little to no effect in clearing compared to C. virginica that had not been exposed. The control decreases slightly after the first initial spike, but then continues to increase. The control is less affected by the Karenia brevis because it has not been previously exposed. The control is again shown with the highest clearance rate in liters per hour, initially at about 0.339 liters per hour and increasing to about 0.680 liters per hour. Figure 3 shows the average clearance rate. The averages of each trial show the overall trend that the previous exposure to K. brevis significantly decreases the filtering ability of the oysters. The length of time the oysters previously exposed does not seem to make notable difference, whether it be 1 day of exposure, 2 days of exposure, or 3 days of exposure. The percentage change between the control and 1 day of exposure shows a 58% decrease in filtration, followed by a 48% decrease in 2 days of exposure and a 50% decrease in 3 days of exposure. The results support the initial hypothesis that the prolonged exposure to K. brevis would decrease C. virginica's clearance rate. The percent change in clearance rate of K. brevis between the control and 1 day of exposure showed a 58% decrease in clearance rate. This decrease represents how significantly the oyster's clearance rate is affected after initial exposure to K. brevis. The percent change in clearance rate of K. brevis between the control and 2 days of exposure showed a 48% decrease in clearance rate, and 3 days of exposure showed a 50% decrease in clearance rate. Collectively, the data shows that the exposure to K. brevis decreases the clearance rate of C. virginica by an average of 52%. This data supports previous research that brevitoxins produced by K. brevis have a negative effect on marine organisms when exposed. Because the clearance rate of the oysters is affected significantly, this may contribute to the recurring blooms along Florida's coast and other areas. A mature oyster has the ability to filter up to 50 gallons of water a day, and with such an abundance of this organism, these filter feeders are an essential part of filtering the ocean's waters. With initial exposure to the bloom, the oyster's ability to filter K. brevis out of the water is decreased by over 50%. This also affects these oysters' ability to filter out excess nutrients in the water. These blooms are then able to grow and flourish in an environment where the weakened oysters are clearing less and less K. brevis and nutrients. So while the initial clearance rate is affected significantly, the data shows that prolonged exposure does not further decrease the clearance rate. After 3 days of exposure to Karenia brevis, C. virginica maintained a clearance rate of about 50% of its initial filtering capability. This observation of the data is extremely important because it shows that C. virginica still has a lasting ability to filter when continuously exposed to K. brevis. Compared to other organisms, this may be a viable candidate in red tide mitigation. C. virginica may be used in abundance to increase efforts in diminishing K. brevis concentrations during red tide blooms. Due to limited resources while testing, only 2 trials were carried out in experimentation. These 2 trials were not enough to perform a statistical test to test the statistical significance between the trials. However, when statistical analysis was conducted to compare the days of exposure, no statistical significance was found between 1 day of exposure, 2 days of exposure, and 3 days of exposure. The p-value was less than 0.05%, or was greater than 0.05. This supports the previous idea that C. virginica is not further affected by Karenia brevis after initial exposure. This again reinforces the possibility of these oysters being used to filter K. brevis during the red tide blooms. The results support the initial hypothesis that prolonged exposure to K. brevis would decrease C. virginica's clearance rate. The gap to be filled in this area of research was to understand the effects of K. brevis of the filtering capability of C. virginica in a laboratory environment. This gap was filled with the research done in this experiment, which may be used to carry out further investigation on a wide-scale study. The gap filled in this research details the effects of Karenia brevis on the filtering ability of C. virginica. This knowledge may now be applied to possible applications in solving the frequent red tides affecting areas containing these oysters. A possible application of this research may be the start of an aquaculture industry to produce C. virginica for distribution in areas repeatedly affected by red tide blooms. Many C. virginica filtering K. brevis from the water may help to control the size of red tide blooms before they can reproduce rapidly. Additional application of this research may be the protection of the C. virginica species. This research has shown that the oysters has a lasting capability to filter K. brevis from the water after initial and continual exposure. With this gap in the research filtered, it is now known that even when these oysters are affected by the red tide blooms, they are still able to filter out K. brevis at a rate of about 50%. In an area where a bed of C. virginica are abundant, boundaries may be put in place to protect from boating, fishing, and other recreational human activities. As far as limitations in the research, these occurred with limited laboratory space. There was also a limit in the supply of C. virginica that was available from local areas without disturbing populations. Karenia brevis was also limited from the amount that was available in Molt's culturing department because of other experiments that were being performed at the time. A possible source of error could have been the brevetoxins aerosolizing and affecting the control tank. And while this is unlikely, it is still possible because the tanks were in the same room. Excessive aeration may have also affected the exposure dose in treatment tank by killing Karenia brevis. Cell counts may vary due to algal deposition and sample to sample. It doesn't stay uniform throughout the tank. And also, oysters collected from Sarasota waters may have been previously exposed to low concentrations of Karenia brevis. For future research, the results of this experiment show that despite the decrease in filtration rate, C. virginica is still an organism that is capable of filtering K. brevis when exposed. This experiment may be carried out in a large-scale study, like previously mentioned, to further confirm results. Different periods of exposure did not present a further decrease in filtration rate. This may make C. virginica a viable source for red tide mitigation. Future research supporting the same conclusion could lead to a species-specific protection of this organism in local waters. This would ensure C. virginica remains plentiful in the area to filter blooms of red tide instead of removing for harvesting or other reasons. Implementation of this species of oyster in problem areas of red tide may also be a future option to help aid in red tide mitigation. Future research would include investigation on the ability of an abundance of C. virginica in ocean waters to filter a red tide bloom. And these are my references. Thank you.